Hello, everyone, and welcome to today's webinar, Circulating Inflammatory Biomarkers Associated with Cognitive Function and Dementia. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink Proteomics. To learn more, visit olink.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Dr. Margaret Doyle, Associate Professor of Pathology and Laboratory Medicine at Larner College of Medicine, University of Vermont. For a complete biography on our speaker, please click on the Presenter tab. Dr. Doyle, thank you for joining us. You may now begin your presentation.
Thank you, Kaylee, and thank you, LabRoots and Olink for hosting this webinar. I'm excited to present to you today some of the work we've been working on looking at circulating inflammatory biomarkers and their association with cognitive function and dementia. I do have some non-relevant disclosures here. And so the work I'm going to present to you today is from a paper we've recently published in Aging Cell, titled "The Peripheral Inflammatory Biomarkers Are Associated with Cognitive Function and Dementia" from the Framingham Heart Study Offspring Cohort Study. And this work was piloted by our PhD candidate student, Jia-Chen Chen, pictured here. The goal of this study and why we did it is that we had done some previous studies showing a role for specific circulating inflammatory factors and immune cells with neuropsych testing, MRI, and incident Alzheimer's disease and related dementia. Our main goal was for this grant that we had funded was to look at the role of the immune system and incident dementia and Alzheimer's disease. But because of delays with the pandemic of getting the immune cells actually phenotyped, we were looking at proteins that had previously been measured in the Framingham Offspring study. And because of that, we were finding that there were proteins that one would not normally think of when thinking about Alzheimer's disease that were showing associations with neuropsych testing and MRI data. So we started asking the questions, were there other proteins involved in inflammatory pathways, and particularly ones that were not previously investigated in the dementia, Alzheimer's disease area that would associate with the domain scores that had been developed from neuropsych testing and or incident dementia or Alzheimer's disease. So with this in mind, we first took our Framingham cohort participants, and these are the people that attended the Offspring exam number 7. So the Offspring cohort are actually the children of the original Framingham Heart Study participants, originally recruited in the late 1940s. So this was their exam 7 cycle because they come back every few years for another exam that were collected between 1998 and 2001. And during that exam, a subset of participants had peripheral blood mononuclear cells saved. And since that was the primary goal of our grant, we chose people that had two vials left so that we weren't using them all up. We then excluded participants that were under the age of 40 or individuals that were already had dementia at this exam. And then we prioritized for people that had a significant amount of follow-up, so that we had 1,000 people in our primary cohort. From there, we lost 4 people that they did not have sufficient cells in their tubes for immune cell profiling. Then we further looked at what we had for plasma samples within this group, and we had 879 participants that we sent off to Olink for analysis. For our study, we excluded 2 because they failed QC in the biomarker run. We took out 8 because they were missing ApoE genotype data. And then we had further exclusions depending on whether their neuropsych testing was within 5 years of the exam or whether they had prevalent stroke or missing covariate data in our Alzheimer's disease outcome. And so these are the basic demographics of the two groups that we looked at. We had the neuropsych factor score testing sample and the dementia sample. And as you can see, they are pretty similar cohorts. So we felt good about moving forward with looking at these two because they're subsets of each other. So we, as I mentioned, we had sent sample to Olink in Boston to have analysis of these proteins in our sample. The Olink uses, we chose to target 96 inflammation panel because inflammation is what we were most interested in. If you're not familiar with the Olink technology, for each protein, there are 2 complementary antibodies that bind on different portions of the protein. And attached to each of these antibodies are strands of DNA that are complementary to each other. They will only cross-link when they are both bound to the same protein. We then use those cross-linked proteins to generate barcodes and amplify the signal, and then the readout is either by qPCR or next-gen sequencing. As you can see here, it goes through the strengths of the platform. What it does use is very little sample volume, which is a strength to give you, for this panel, you get 92 protein, plus the other 4 proteins in the panel are reporter proteins for QC purposes. But it's a very specific assay, and it has a very wide dynamic range of measurement. So out of these 92 proteins, we made the decision in our group to only use proteins for our analysis that had over 50% of the participants had values over the lower limit of detection. So of these 92, we had 68 proteins that met this criteria. And so here is the mean and standard deviation for these proteins. We should note for these target 96 panels that the data that you get is a normalized protein expression, which Olink calls an NPX unit. So you're measuring relative quantification, and note that it is on a log2 scale. So 1 NPX different is indicating a doubling of protein concentration. So for our analysis, we had taken, because at every exam and for different studies, people are utilizing different neuropsych testing platforms. So a group of people from different cohort studies came through, and they developed domain score, factor scores for each of the different domains based on what type of testing and what sort of measurements they were getting, so that we could harmonize data across different exam years and across different cohorts. So using this, we developed scores for the memory domain, executive function, language domains. For the visual domain for Framingham and exam 7, the only test we had that was part of the domain was the Hooper Visualization Organization test. So that is the only test that is within the visual domain for this analysis. The protein levels and the neuropsych testing were all inverse normal transformed. This reduces skewness, and it gives us a distribution with a mean of 0 and a standard deviation of 1. We did this mostly because it makes our effect sizes so they're all on the same scale, so that it's easier to visualize. We also went through and we wanted to look, one of the main things we wanted to look at that we didn't see happening a lot in the literature is to look at people by their ApoE genotypes. We know that ApoE4 has been associated with an increased risk of Alzheimer's disease, and ApoE2 alleles are associated with a lower risk of incident Alzheimer's disease, whereas the ApoE3 is the common genotype. So we used additive coding for the combined analysis for the E2 and the E4 alleles. And when we stratified, we did not include individuals that were ApoE2, E4. And then we considered ApoE2 to be the E2, E2s or the E3, E2s. and E4 carriers were E4, E4, or E3, E4. And generally, the ApoE3 genotype was our reference value. So our primary models, we included sex, age, education level, the time between the testing and exam 7, and a retest indicator, because we know that generally the first test gives different results than the second test, because the second test, they know what's coming a little bit, so they tend to do a little better, and the ApoE genotype. For model 2, we included the cardiovascular risk factors, our cardiovascular disease, and we also included risk factors for cardiovascular disease. Because this was a very exploratory analysis we were starting out with, we used a false discovery rate of less than 0.1 to account for multiple testing. So in our first analysis, we were looking at the executive function score. And I apologize, this is small and somewhat difficult to read, so I will kind of walk you through it a little bit. So these are the proteins that we found that had any kind of a positive, a significant association with a false discovery less than 0.1. What you're seeing on your x-axis is the effect size, again, because we set them up so they would all be at 0 with the mean. This is, our scale is going from a -2 to a -0.2 to a +0.2. We have, for each person, there are 4, or for each protein, there are 4 different graphs. The first one is the pink scale squares, and that is everyone. The blue circles underneath, those are your E2 carriers. And again, remember, E2 is good as far as Alzheimer's disease. The pink triangles or the red triangles, those are the E4. Those are the ones that are increased risk for Alzheimer's disease. And then the green diamonds are the E3s, and that's the majority of the population. So the first thing, when you look quickly down at the pink squares going down the list of proteins here, you see that for the most part, they are going, and they're listed here from the most negative effect size to the less negative. And you see that what we're seeing is that the majority of these proteins are on the negative side. What this is showing us is that like a higher IL-6 for the first one means you're not performing as well on these executive function scores testing. And so we see for the majority of the proteins in the inflammatory pathways, higher levels are not good for your executive function testing. So when we kind of zoomed in, because there was a lot of data there, I zoomed in on those that had an FDR less than 0.05. So these are the most significant things we saw using our model 1. And as you can see, for a lot of these, if you like take the IL-6, for example, if it was significant for everyone, it was also significant mostly in the E3s. And that shows that the significant function is the general population. So with the IL-6, and you see that the effect sizes are very similar between all and the E3. So it's driven by the normal population. Then you see things like the FGF21 there at the top, and that is only in the E4s. But if you have, so if you have higher of FGF21, which is a fibroblast growth factor, you're going to perform less well. To me, things that were most interesting here was, though most things were negative, we did see 2 that had positive effects. So for PD-L1, which is an immune checkpoint inhibitor, higher levels of PD-L1 in the E2 carriers had higher executive function. So this may be indicating to us that there's something by inhibiting the immune checkpoint. So with the immune checkpoint inhibitors, they stop your immune system when it starts to over ramp up. So that the fact that in these E2 carriers, it is higher levels of PD-L1 is good for your executive function. It may indicate that there is something that is affecting your executive function, that having this immune checkpoint inhibitor higher levels of it and blocking the immune system is what is giving you better outcomes in an E2. The other one I found very interesting was this tweak, which is only in the E4s, which we know E4 is a higher risk factor for Alzheimer's disease. But in these people that are carriers of this E4, if they have higher levels of tweak, they are performing much better on the executive function testing. And so sort of zooming in on these associations, you can see that that they're actually the E2s and the E4s are going in pretty much opposite directions, although not always significant. And when we go ahead and then we bring in the model 2, where we are now adjusting for prevalent cardiovascular disease and cardiovascular disease risk factors, we see the only thing that remains significant is the PDL1, the immune checkpoint inhibitor, again pointing us to something in the immune system that may be affecting the executive function scores. When we further moved on to language scores, again, it's the same E2, E3, E4, and all here that we're seeing. And we're seeing the only significant associations are with all of them, and in general, all of the subtypes look very similar to the main group. So we see that, again, they're all in the higher levels of the inflammatory proteins, worse performance on the testing. When we bring in model 2, we lose the significance on this trans protein. The IL-10 and the LIFR were not affected. And we also brought in a new significant protein, which is the CCL19. It's thought to be an angiogenesis suppressor. And we see that this is, again, only in the E2s, and higher levels of this in the E2s are showing a worse performance in our language score domain. Moving along to memory scores. Again, we see things that our only associations in the memory score are in the E4s that are significant at this level. No, I said that. Yeah, that's correct. So again, very interesting. We see the tweak going in opposite directions for the E2 and the E4, which may indicate this is a very important protein in figuring out why E2s are protective and E4 is a risk factor. We're sort of seeing similar situation in the DNER, which is a delta notch-like epidermal growth factor related receptor. And this regulates interferon gamma secretion. And the IL-17, we're seeing that it is much lower in the E4 carriers. And again, when we bring in the prevalent cardiovascular disease and risk factors, we see that there is not much change, although nothing in the effect sizes, but nothing rises to the level of statistical significance. And again, looking at the visuospatial scores, we're seeing the only significance that we're seeing here is in the E2 carriers. And again, it's all higher levels of these proteins are associated with worse visuospatial scores. And when we adjust those for cardiovascular disease, again, in model 2, we see not only do these same things remain significant, but we've also increased and we found many more proteins that were also significant when we're adjusting for the cardiovascular disease risk factors. So next we went ahead and we're looking at incident all-cause dementia and incident Alzheimer's disease. And so for this one, our x-axis here is our hazard ratios. And so we're seeing an increased risk for TNF-beta in the entire sample and plus our E4s and our E3s. Note that here we do not have E2s because we did not have a significant number of cases of either dementia or Alzheimer's disease, because remember, these are the people that seem to be protected from it. And we're seeing that in our sample of people followed for almost 20 years, that we did not have enough to do the analysis. So again, the two main things that we saw, the most high hitting ones, are the TNF beta and this CDCP1, which is a cub domain containing protein 1. And they were similar for both the E4s and the E3s. For many of the rest of these, we saw only associations in these E2s. And again, they surprisingly were an increased hazard ratio for E2s that have high levels of these proteins. When we looked at incident all-cause dementia, the TNF beta was the only protein that came up. And again, it was only in the E4 domain. And again, when we brought in model 2 and we adjusted for prevalent cardiovascular disease and risk factors, we saw that we lost significance for a lot of the proteins. But again, the TNF beta remained significant. The cub domain containing protein 1 remained significant. And in this CX3CL1, we see significance only in the E3 carriers. When we looked at the incident all-cause domain, Alzheimer's domain dementia, Alzheimer's disease dementia, we can see that the TNF beta again remained significant. We were no longer significant for the E4 carriers, although the effect size is very similar. It's just the confidence domain, the confidence intervals got a lot larger in this subset. But what's really interesting is that the tweak protein for incident Alzheimer's disease, although only significant in the entire sample, was again showing a protective role. And although not significant, it is especially protective in the E4s. So in trying to figure out what some of this meant and if anyone else had actually done any of this before, I started coming through the literature and I found this really interesting paper. And why it was interesting was this group was originally looking at diets, and they had looking at diet questionnaires. They had found the different types of diets that they based on their eating questionnaires, but then they looked at them with, again, a larger panel of O-link proteins. And what they were finding was that, so they found these proteins that were related to what they were calling an inflammatory diet. It was something that they were predicting would cause inflammation if you ate it, primarily ate this type of a diet. And then they went further to look at, do these proteins that were related to an inflammatory diet predict future cognitive impairment? And they did this in 3 different studies. So on the left panel, you see the Women's Health Initiative memory study and the Esther study. And that you see that proteins that were in our panel that came up were like the CDCP1, and it was significant for both of the individual studies and when they meta-analyzed the study. Hazard ratios very similar to what we saw. And they also saw the CCL3, which we saw in our analysis. Then they went to replicate their studies in the ERIC study. And again, they saw the CDCP1 was significant. And here they split it by male and men, or women plus men, or women. Okay. And we see that there was no real difference between the male and female, but that again, it is predicting incident dementia, cognitive impairment, but their CCL3 was not. Note that also that the ERIC study is recruiting for atherosclerosis risk. And so we did see that the CCL3 in our study was no longer significant when we adjusted for cardiovascular risk factors. And then in looking at things in a different way, this paper by O et al. Was looking at differentially expressed genes in cognitively normal persons with mild cognitive impairment and people with Alzheimer's disease. and looking at the differences in the genes expressed. And out of these genes that they found, we see, looking at cognitively normal versus mild cognitive impairment, we see that the CCL3 came up, which is also in our studies. When they looked at the cognitively normal versus Alzheimer's disease, we see the TNF beta there as also shown up. And again, looking at mild cognitively impaired versus Alzheimer's disease, we see the CCL3 again, which was in our studies. So this was somewhat of a validation of some of the results that we found that were found in different manners, but showing some of the same pathways that we were looking at. So looking at some of the strengths of our study is that we leveraged our community-based Framingham study that's been deeply phenotyped for cognitive aging with regular neuropsych testing, and it's well characterized for cardiovascular disease and risk factors. They were also followed for over 20 years following. So we started with dementia-free people, and we followed them for 20 years with standardized testing and a dementia review administered by standardized protocols. We have stored plasma samples at exam 7, allowing us for the comprehensive profiling using the Olink panel. But we also have the neuropsych testing very nearby. We have MRI data very nearby. We're also doing the immune cell phenotyping. So we're trying to get a big picture of what's going on with cognitive aging. We also have ApoE genotype status on our cohort, which allows us to look at these ApoE carriers to try and find, again, significant pathways for the different types of disease that we're seeing going on with everyone. There are, of course, limitations to our studies. We don't have a huge sample size, 1,000 people, and then we lose some along the way. And this particularly made it difficult to see things in the E2 carriers when we're looking at incident all-cause dementia and AD sample dementia outcomes. It was cross-sectional, so you cannot, for the most part, with the domain scores is cross-sectional, and you can't use it to infer causality. There is variance across the domain scores, so we may not be effectively looking at individual performance within a domain. The blood draws for the biomarkers and the neuropsych testings were not done at exactly the same time, but that's why we limited our testing to samples that were within 5 years of each other. There's always a possibility that the inflammatory cytokines may be degrading with time, even at -80, and that the time difference in storage across the participants, because there is differences, may create differences in protein levels that are hard to detect. We measured our inflammatory proteins at a single time point so that, again, we may see more if one looks at changes over time. We also know that the circulating protein levels may not be reflecting at all what's going on in the brain, and that probably the most significant limitation is that the Framingham offspring cohort participants are primarily white, well-educated, and reside in New England. So replication in independent samples that are ethnically and geographically diverse are required to confirm our findings. And so in summary, several circulating inflammatory proteins have been found to demonstrate an association with the cognitive scores. The stratified analysis are suggesting different protein effects between the E2 and E4 carriers, with most E4 carrier associations with the executive function and memory domains, and most of the E2 associations are with the visuospatial domain. Higher levels of TNF beta, a protein known to regulate the NF-kappa beta pathway, upregulating Th17 and be an anti-proliferative protein, and the Cub domain containing protein 1, which is reported to play a role in T-cell migration and chemotaxis and may contribute to autoimmune diseases, were associated with an increased risk of incident all-cause and AD dementia. And higher levels of tweak, which are part of this tweak FN14CD163 axis, which is involved in metabolic disease, chronic autoimmune diseases, and acute ischemic stroke. In ApoE4 carriers, shows a protective effect in both the cognitive immune function testing and incident Alzheimer's disease. And so our future studies are, we are currently preparing manuscripts where we looked at these O-link inflammation proteins and the brain MRI phenotypes that should be being submitted in the next week. We're looking at the role of circulating immune cell subsets with these O-link proteins. We're in the process of developing a risk score that combines immune cells and proteins. We're also looking at association of the inflammation proteins, immune cell subsets, and the neurology protein panels that are very popular these days in potentially predicting who's going to have Alzheimer's disease. These include things like the A beta 4042, the total tau, and the different phosphorylated taus, neurofilament light, and the GFAP proteins. We also started a new program we're looking at with a group of very intelligent people working out of Harvard, where they're taking CT scans from chest and lung CT scans, and they're generating scores based on their lung health and their immune health, which they're quantitating by looking at the thymus. And we're looking at the association of those with the O-link proteins and the immune cells. I find that's really fascinating work, and I can't wait till we get to dig in more. Some of our future studies is looking at replication in other cohorts. We're looking at like Jackson Heart Study because it's very similar to Framingham, except it's an all Black population in the Jackson, Mississippi greater area. MESA, which is a multi-ethnic study. We're also looking at some things we can do in regards because they also have very good cognitive function testing over time in a multiracial cohort. We're exploring the use of a risk profile that would encompass adding immune cells and these O-link proteins and demographics and MRI data so that we can perhaps pull together some kind of a risk profile to help in predicting healthy aging and potentially places where one can affect the outcome of your healthy brain aging, which is why I found the paper with the diet linked to the O-link proteins linked to the incident dementia, a fascinating way to see if there's potentially an area where one could alter your dietary habits that might affect your risk of future dementia. The other thing that would be great to look at is, can we look at the proteins over time and see if we can develop trajectories that might be telling us that it's not so much that you have high tweak if you're an E4 carrier today, but if your tweak levels are going up and down over time, that might be further increasing or decreasing your risk of dementia. And then, you know, the ultimate goal is that can we find specific pathways for when we bring all these results together that would help us to then find ways that we could improve outcomes starting at an earlier point to, instead of trying to correct what is the outcome, which is dementia, can we stop it from ever getting to the point where we're at dementia? And with that, I would very much like to thank my coworkers on this particular grant from the NIA, my co-PIs, which are Joanne Marabito and Kathy Lunetta, our co-investigators, Jesse Mez, Claudia Satsabal, Michael Alasko, and Wendy Q, and our postdocs and graduate students on the project, Yan Feng, Ahmed Ragab, Jachen Chen, and Yumeng Chow. And of course, we'd like to thank the NHLBI, Boston University School of Medicine, the Framingham Heart Study, and all the Framingham participants and all participants in general that make these studies possible. And I'd also again like to thank O-link and LabRoots for sponsoring this webinar. And I am ready to take questions.
Well, thank you so much, Dr. Doyle, for your informative presentation. Before we start our live Q&A session, I'd like to invite our audience to participate in a few poll questions. So our first poll question here asks, overall, how would you rate your webinar experience? So our answers are very good, good, fair, and poor. Again, the question asks, overall, how would you rate your webinar experience? We'll give you a moment to submit your answers. All right. Our next question asks, do you use any immunoassay platform to measure circulating biomarkers for your research? Yes or no? Again, the question asks, do you use any immunoassay platform to measure circulating biomarkers for your research? Give you another moment to submit your answer. Okay, now we have one more question for you. This last question asks, are you planning any biomarker experiments soon? Yes, no, or maybe within 12 months? Again, the question, are you planning any biomarker experiments soon? All right. I'll let you finish your answers there. Okay, great. Well, thank you so much for your participation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here asks, for proteins that are not classically secreted proteins, such as PD-L1, is there a way to distinguish if this is truly soluble PD-L1 versus membrane fragments or exosomes expressing PD-L1?
Well, that is actually a great question, and one that we have often asked here in our lab and are currently working on methods to do that. So I would say probably at this point, certainly not with just using the Olink platform, right? But one would think that you're more likely looking at a soluble form that is not on a membrane or an exosome with membrane fragments, because of the need to have two separate antibodies that are binding it. So in order for the protein to have room for antibodies that are to fit it, the possibility is less likely if there are membrane fragments, because the phospholipids will be sheltering part of the protein. But we are trying to look at, right now, we've developed some methods to look at exosomes specifically. And we've tried for some of these proteins, not these particular proteins, but other proteins we've been interested in the past, to try and separate them by gel filtration and stuff, just to look at what size they are, to see if they, the ones floating in plasma, are actually have any of these phospholipids, because that would drastically alter the size. But that is, again, a great question, and we are all looking into that as well.
Great, thank you. All right, next question. For the tweak protein in the E4 carriers, have you identified potential pathways this protein is linked to? And if so, is there any work being done to consider further investigation?
So we have not exactly found the potential pathway that it's linked to. So as I mentioned, again, and sort of similar to the question before, a lot of these proteins are normally found attached to a membrane or in a membrane. So they're membrane proteins that for now are some reason soluble. And so a lot of the literature on the tweak protein has to do with the membrane bound form. So there's not a lot on what's going on in the plasma. So there's three different ways that these proteins can become soluble. They can be clipped off the membrane by some reason. They can be part of a prepackaged exosome that is the cell puts out to transport things, or they can be part of when the cell breaks down, the membrane pieces break off, and it could be attached to small pieces that can then form a larger microvesicle as opposed to a smaller exosome. But it's also interesting. So when they're soluble, sometimes what they act as is a decoy receptor, so that by binding whatever its ligand is in solution, it keeps the ligand from then binding the cell and performing whatever activation or pathway signaling it would normally do on the cell. So it could be working as a loop to try and remove activation pathways sort of things, so that if there's too much activation going on, it could be a feedback loop to bring it to decrease the activation. So yes, we are trying to investigate further, but we're trying to figure out exactly where we're going with that next. There's a lot to go through.
Thank you. Now, can all these significant proteins enter the blood-brain barrier?
Well, I mean, that's the million-dollar question, is how to figure out which ones circulating actually go through the barrier, or if they're involved in the opening up the barrier. So main ways things get through the blood-brain barrier are, first of all, if there's any kind of damage to the blood-brain barrier, then everything in the blood can get in. But there's also the possibility that they are working on smaller problems with say the junction proteins that are holding the cells together that form the blood-brain barrier. A lot of these proteins we're seeing are like chemokines or chemokine receptors, or like monocyte chemoattractive proteins, MCP ones. And they may be higher because they are working to bring repair mechanisms or something to the blood-brain barrier. But that's always what the difficulty is trying to figure out why these proteins that are circulating in your blood that we're taking out of your arm are somehow affecting your cognitive function. And so the answer is, I don't know, but these are the bigger questions that we are really trying to answer with doing this work.
Yes, thank you. All right, and I want to thank our audience for submitting so many great questions. We've got plenty more coming through, and we do have some more time, so we're going to continue. All right, this next question asks, were you able to compare the plasma A beta 42:40 ratio to some of these neuromarkers?
So that work is in progress right now. And so I'm going to have to tell you, you have to stay tuned and keep an eye out to your journals, because we are working on that. That is one of the things. Our problem is that most of those protein measures were not measured at the same time as these markers. And so that's where one of the things we're looking into is, because we are seeing some associations with the markers at different time points. So we're trying to see if we can find a way to get them all measured in the same sample so that we know for sure. But great question, really. These are really good questions.
Great. Now, Dr. Doyle, this is a two-part question here. Are there any emerging biomarkers beyond traditional ones that shows promise in predicting cognitive decline or dementia onset? And what are the challenges in incorporating them into clinical practice?
So I'm going to say that the challenge, I'm going to kind of take the second one first. The challenge is always incorporating them and finding early enough proteins is to know which, well, I'm not going to answer this well because it's a really good and really complicated question. So I know that there's a lot of work and a lot of stuff coming out now looking at the neurology markers I mentioned, particularly the new phosphorylated taus, the p-tau 217, I think is the new one that we're looking at. But as always, the problem with looking too far ahead and getting too much ahead is that, first of all, when we find these things, because it's such a complicated disease, that there's probably not one biomarker that's going to tell us everything. And that's why I think instead of looking at single biomarkers, it's that we need to start looking more at groups of biomarkers to try and figure out more which direction we should go. And so I think of this sometimes like with the genetic studies where we found, like for breast cancer, and we found the BRCA genes, and we found ways to treat people that had the BRCA gene mutations. But that accounted for such a small portion of people with breast cancer, right? I think we're going to find the same thing when we're looking at dementia and Alzheimer's disease, is that they are coming by different pathobiology pathways that are affecting different portions of the brain. And that's why I think that when we look at part of the neuropsych testing and the domain scores, is that we know that different portions of the brain are kind of responsible for different types of brain function. So the executive function is coming more from your frontal lobe, whereas your memory and language skills come more from the temporal lobe. And so then we start pulling in the amyloid plaques in your brain. Does it matter exactly where these amyloid plaques are locating in your brain? And are they getting there by different pathways? So I think that while we're getting closer and closer to cracking a lot of the dementia and Alzheimer's disease pathways, there's still so much further that we need to go, because we're going to start finding, as we have with cancer, as we have with heart disease, that there's so many sub diseases within these big broad categories that we're using to classify things right now. And so I think that's the direction we're going. And the incorporating into clinical practice, unfortunately, it takes a lot of time to figure out exactly what we need to do to get the right diagnostic or prognostic tool, and even longer to find out a way to stop it, right? To stop the progression of the disease.
Yes. All right. Next question here. Have any of the protein biomarkers identified here been identified in ARIC or other population-based cohorts, which also looked at incident dementia? Or were these not measured on the platforms used or not significant?
So I did show the one study I noticed with Eric there that had the Cub domain containing protein 1 that was significant in both our measurements and Eric. And the CCL3, which was not, and was in Eric and was not in our study when we adjusted for the cardiovascular risk factors. Again, a lot of these proteins, the little bit of data I did find was using the Olink platform. I know that a lot of these bigger cohort studies in the US are currently, as part of the NIH TopMed program, going to do the larger Olink platforms and across time points. So this will be an area that will be great for future studies to, first of all, verify our data, but also to potentially find larger data, because it's a lot more proteins to look at. So there's a lot of work to do. Always looking for people to help out if they're interested. And yeah, so we're looking to answer a lot of these questions in the near future.
Great, thank you. All right, next question here. What are the pathophysiologic differences between ApoE2 and E4 carriers?
So this is another great question. We know that ApoE is, in general, it's an apolipoprotein. And so the differences between E2 and E4 are very specific site mutations on these on the ApoE protein. Exactly what the differences is, I have not had the chance to really dig into this enough to figure out the exact difference. But that's kind of where we are right now, is we have very big differences, and we haven't seen a lot of papers out there that people have looked at exactly what the E2 and the E4 difference is and how these different proteins might look into it. So this is where I'm finding it's a very exciting area that I'm happy to have more people working on to see what we can figure out these differences.
Great, thank you. All right. Next question here, are there specific inflammatory biomarkers that shows a stronger correlation with cognitive decline or dementia risk? And if so, what implications does this have for early detection and intervention strategies?
So I would say that we saw cognitive decline dementia risk. So what we saw, okay, so there are previous reports where obviously we've looked at as far as the inflammation pathways, things like IL-6, soluble CD14, and they do have really strong correlations with cognitive decline. I could also say that if you look at our data that had stronger correlations with cognitive decline and dementia risk, a lot of them, again, were in chemokine pathways or very specifically in the TNF pathways. So we had both TNF alpha, TNF beta, and a lot of the TNF type receptors. That tweak is also a TNF. It's the tweak comes from TNF superfamily like protein that induces weak apoptosis, which was just how it was first discovered. But so that we're seeing some definite inflammatory pathways involving TNF alpha and beta and TNF receptors. We're also seeing strong signals in a lot of these chemokine pathways. So we need to, again, we need to dig into this more. And if there's basic scientists out there, this is an area that the epidemiology is good at finding these potential pathways. But we need the basic scientists to come in and try and help us to figure out exactly how these pathways are working. So it's a big process with a lot of work to do still.
Yes. All right. Well, that will conclude our session for today. So Dr. Doyle, thank you. Do you have any final comments for our audience?
Well, I'd just like to thank again LabRoots and Olink for letting me discuss our results. I'd like to thank you all for coming and for the great questions. And as you can see, this is, as an exploratory analysis, it gives us some ideas and some pathway ideas, but there's still a lot of work to do, and there's a lot of room for people that come into the space and help out because it's an important disease, and we would really like to, try and figure this out and work on better treatments because there aren't a lot out there. Thanks.
Well, thank you again, Dr. Margaret Doyle, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Olink Proteomics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.